My name is Scott Lewins. I'm a entomology extension educator with UVM Extension. I work with the Northwest Crops and Soils team. You'll be hearing from one of my colleagues at the end of this session. But first, we have Dr. Jessica Lubell-Brand. She's an associate professor of horticulture in the Department of Plant Science and Landscape Architecture at the University of Connecticut. She has 20 years of experience working with the horticulture industry. Dr. Lubell-Brand has conducted research with hemp on feminized seed production by masculinization, phenotypic and genotypic analysis of selfed and outcrossed feminized seed, production of polyploids, and tissue culture micropropagation. Dr. Lubell-Brand is going to be talking about creation, performance, and benefits of triploid hemp. And so I will turn it over to you.
Before I get started, I just want to acknowledge the people that have worked in my lab who have helped me develop the research that we'll be presenting today. So the first is Lauren Kurtz. She's received her master's degree in my lab, and she's now working towards a PhD. And the second picture is Mark Brand, who's a colleague of mine. And then I've had 3 undergraduate students work in the lab, starting with Carl Johnson, and then Jessica DiMatteo, and currently Lillian Borbis is an undergrad in my lab doing some research. So before I get into details, I want to just define what ploidy is, which is the number of sets of chromosomes that an organism has. And every organism has a base number that we symbolize with lowercase letter n, some base number of chromosomes. So in humans, for example, we are diploid organisms because we have 2 sets of chromosomes, and a set contains 23 chromosomes. So when a new individual is created, 1 set of 23 chromosomes comes from the father, 1 set of 23 chromosomes comes from the mother. They fuse for the offspring to have a total of 46 chromosomes. Those 23 chromosomes from each parent, they actually correlate to each other by number, and they're called homologous chromosomes. So if you're interested in genetics and breeding, you know that homologous chromosomes carry the same genes, they have the same amount of DNA, and they're the same size. So like humans, many plants are naturally diploid, containing 2 sets of chromosomes, including cannabis. In cannabis, both the male and the female gametes, or the egg and the sperm, contribute 5 chromosomes. So the N equals 5 for a total of 10, or 2N equals 10 in the new diploid organism or embryo that's inside the seed. So the pictures here are just showing, when pollen reaches the female flower and fertilization occurs, that's fertilization, and then that's pollination. And then when the egg and sperm unite, that's fertilization. And it results in a viable seed when everything goes well and according to plan. And the parents are compatible. Many plants in the plant world, unlike the human or animal world, are naturally polyploid, which means they have 4 sets of chromosomes, 6 sets of chromosomes, 8 sets of chromosomes, 4N, 6N, 8N. Like many conifers are hexaploid. They have 6 sets of chromosomes. If you're a gardener or are into landscaping and you're familiar with the butterfly bush or summer sweet called clethra, these are natural tetraploids. If you're not familiar with those, you're probably familiar with the fruits like strawberry, which frequently the ones that we eat are hexaploid or octaploid. And some genera, like clethra, for example, have varying polyploidy among the species. There's a dodecaploid clethra, I think. Frequently, but not always, polyploids are more robust than their diploid counterparts. And in strawberries, frequently the octoploids, like you see here, are bigger, they have bigger fruits than the diploids. And in daylilies, daylilies is a good example. My background is in ornamental plants. So tetraploid daylilies have bigger flowers, bigger buds, you know, thicker stems than diploids. When you have an even number of sets of chromosomes, like 2N, 4N, 6N, you frequently have fertile organisms. They can fuse their gametes and produce viable seed. They may have varying fertility rates between the ploides, but usually within reason. Fertility is okay, and it happens when you get really higher. The higher you go, of course, it's a lot more DNA to copy, and so perhaps it doesn't happen as frequently successfully. So that'll become more clear as we move on. So you've probably heard of seedless watermelon. You've eaten it. You've had seedless citrus. If you're into the Cannabaceae and you're into hops, you've maybe heard of seedless hops. These seedless crops are triploid, which means they have an uneven number of sets of chromosomes, and we abbreviate them as 3N. Triploids frequently do not produce viable gametes. They don't produce viable egg and sperm, so they don't set seed, certainly to the degree that a diploid or a tetraploid counterpart may. So the process of meiosis, which produces the sex cells or the gametes, another term gametes or egg and sperm, is disrupted when you have an uneven number of chromosomes in a parent. If we could make seedless cannabis, it could solve the problems of pollen drift from fiber and grain farms, or that random male plant or male flowers that can just show up in a female crop and can travel for many miles. We know that this has been a problem in the industry, in the hemp industry and medicinal cannabis industry, as you can see here by these newsreels. We know that when a female flower produces seed, that flower is rendered unsaleable. And it also reduces yield and cannabinoid production. But you know, for the cut flower market, a seeded flower is just unsaleable. So this is a problem, and for many. A triploid, theoretically, if it were hit with pollen drift, it would not set seed. So natural triploidy is pretty rare in the wild. It can occur when you have a plant species that is both tetraploid and diploid, and those tetraploid and diploids are growing together. You can get natural triploids. But you can also develop them in the lab or in the research greenhouse by crossing tetraploid and diploid plants. And this is how we got seedless watermelon and seedless citrus and seedless hops, by plant breeders creating this cross by syncing up the diploid and the tetraploid, forcing them into flower, and sometimes hand pollinating or just facilitating pollination and all that. And this is showing an example with watermelon. So as I mentioned, cannabis is naturally diploid. So in order to make a triploid, we first had to make a tetraploid. We had to double the number of chromosomes or double the DNA in a diploid plant to make a tetraploid plant. My lab utilized a method called using pre-germinated seeds. and the chemical, which is actually comes from a plant called colchicine. So there are other published methods for making tetraploid cannabis, but we use this method where we take we call a pre-germinated seed, where just the radical is sticking out, and we submerge these seeds in a colchicine solution. The colchicine is a mitotic inhibitor. So in order for this radical to have grown, and for the shoot apical meristem to grow, it requires cells to divide, copy, divide, copy, divide, copy, divide many, many times. That equals growth. So that process of cell division is called mitosis, and the mitosis results in growth. So if you hit an actively dividing shoot apical meristem with colchicine, the DNA can double, but the cell can't split. So you seize the splitting of the cell and you trap the tetraploid status of the chromosomes. So figuring out the right concentration of colchicine and the right duration of exposure of colchicine is important. It's a species specific kind of thing. And this is a lot of the work that Lauren did for her master's degree, part of it, figuring out an optimal rate and duration of exposure to colchicine using pre-germinated seed. And so other labs have done this using different methods. So there you can use a seedling and put a bead of colchicine solution on the shoot apical meristem. You can use in vitro plantlets. These are a couple of other papers you can find in the literature. And then we kind of grow out the seed, and some of them die, and some of them are unaffected, and some of them are chunky and slightly distorted, and that's usually an indication that we've messed things up, and that might be a good one. We test the ploidy of all of our colchicine treated seedlings using flow cytometry, which is a way of measuring the DNA content. We take leaf tissue and we chop it up in a buffer, and this is just the nuclei, so the nucleus within the cell. The nucleus contains the DNA. And then we put this in the flow cytometry machine, and we get a readout like this, where the peak represents the DNA content, and the positioning of the peak represents whether you have the diploid condition or the tetraploid condition, which is twice that of the diploid. And of course, this would be what we want. Sometimes we get mixoploids, which have both cells that were doubled to form tetraploid and cells that were unaffected and remained diploid. And that's actually called chimera. And this picture here is identifying a chimera. So if you will, let's imagine all of these white cells in the shoot apical meristem here. So this is like the little plant. Here's the shoot apical meristem. All the white cells were converted to tetraploid, but some of the cells remain diploid. And this would not be a good breeding parent, because it's not going to give you reliable results in the offspring. So mixoploids we cull, we don't keep. So we successfully made a tetraploid, and we crossed that tetraploid with a diploid and got a triploid. And we were the first to report triploid hemp and a method, our pre-germinated seed method for making tetraploids. And this was in 2020. And this is an open access paper in Hort Science Journal. So you can, if you want to read more about this, you can. And you can see that the flow cytometry peak for the triploid was intermediate between the tetraploid and a diploid, which is exactly where you would expect it to be. And these were some of the early triploids. They were one strain. They were a wife tetraploid by a wife diploid. At the time, My licensing was very restrictive in what cultivars I can use. So we had a wife by wife triploid. It was a sibling cross, slightly inbred. And so, we babied things along and showed it. We still have these triploids, actually. We've kept them going through vegetative propagation. So after that, we wanted to conduct what's called reciprocal crosses, crossing where you have the diploid as the mother with tetraploid pollen, and the reciprocal using a tetraploid mother and diploid pollen to investigate if one direction or the other is better for large quantity production of triploid seed, like for sale. Not for us to sell, but for a grower who might be interested in doing this. The reciprocal crosses had to be done at separate times to avoid pollen contamination. And we used only feminized pollen in our crosses through masculinization using silver thiosulfate as a foliar spray. Silver thiosulfate is in the literature back from the late 1970s and early 1980s. It has been shown to masculinize female plants so that they produce pollen. We amended the method using foliar sprays and published a protocol for that. This is just how we do things, and there are multiple ways of doing this, but we initiate the first spray on the first day of short days for a photoperiod type. We use a 3 millimolar concentration of silver thiosulfate. We spray every 3 to 5 days for around 4 weeks, which usually results in 5 or 6 sprays total. Of course, this is genotype depending because some reverse or masculinize a lot easier than others. Some put out lots of dusty pollen, some don't. And once we see the male flowers are well developed and the pollen sacs are visible, we stop spraying just prior to anthesis or pollen release. We store pollen in the freezer. We've stored it for many months and still have it work. And this is just kind of showing you these pictures Lauren took, showing kind of the progression or the development of male flowers on female plants. There are challenges to feminized seed production. A lot of these are being overcome. Early on, I think there was pollen contamination. If you saw males in feminized seed, it was probably the result of pollen from a male plant getting in. You just didn't have your plants isolated enough. We see a lot less of that now. You also want to avoid using monoecious genetics. Certain environmental factors can trigger the formation of male flowers on female plants, but some varieties, cultivar strains, whatever you want to call it, make male flowers more frequently than others, and you have to watch for those. You have to slough these off, remove these male flowers. This is non-target pollen. We don't typically see strong phytotoxicity on the foliage. We try to spray and have any supplemental lighting be off. This is the worst damage that we've ever seen, and these plants were growing in a very cold greenhouse. At our research farm, we don't use that location anymore, but we were trying to isolate. And but I hear there can be phytotoxicity. I'm sure it's a strain dependent thing, but this is not something that has held us back at all with this process. Okay, so I'm going to present some of the results of our crosses very quickly. And this table looks overwhelming, but I'm going to break it apart, break it down now and explain what's going on. The text box at the bottom, the color text, see it's a black text, that corresponds mostly to the black data in the table. So the text color will change, and we're going to move around and come back and forth to this table. But just to orient you, this first column is the mother cultivar, the ploidy of the mother, the paternal cultivar, the ploidy of the father or the pollen source, the cross notation, the mother always goes first, the number of maternal plants that received pollen. This is the seeds per plant. These are the number of brown or viable seeds per plant, the number of tan or white non-viable seeds per plant, percent viable. We did some germination tests, and we've done some flow cytometry, which you see here, and then some kind of comments, which just facilitates my discussion here overall. All right, so the first cross we set out to do is we had diploid mothers and we had tetraploid pollen from the cultivar Kentucky Sunshine. We had things looked good to start. It looked like the diploid mothers were making loads of seed, but as things progressed, we were noticing that the seeds were not ripening. They were not turning brown. And when we pulled them off the plant, they were hollow inside. In the end, we got thousands of seed per plant. For wife, we got over 3,000, almost 4,000 seed. For a couple of abacus plants in there, we had, close to 800 seed. And for this tangerine, we had high seed as well, but they were largely unviable. There were only less than 1% brown, healthy, filled seeds on this plant. And what we think is happening here is that the endosperm inside the seed is failing, so the embryo doesn't grow. So this is showing you on the left, brown, healthy, viable seed, and on the right, tannish, hollow to deformed seed that is not viable. And so the majority looked like this. We did see triploids in the viable seeds. We saw some diploids in the viable seed for these two cultivars. We had, my box is covered here, but wife is one of those cultivars that loves to throw up male flowers. So we believe that these diploids resulted from pollen contamination. We did have a tetraploid mother in this greenhouse section at the time of this experiment, and it set a lot of seed. This was about a 3-foot tall by 3-foot wide plant, and a large amount of those seeds were viable. So this cross worked, but the 2N by 4N crosses really did not work at a commercial scale for sure. So what we think is going on, in order for me to explain what I think is going on, I first need to explain to you what double fertilization is. Double fertilization is when the pollen grain lands on the stigmatic surface, it builds a pollen tube, and the generative nucleus falls into the pollen tube, and that forms two sperm. 2 haploid or N sperm. One of the sperm unites with the egg, creating the traditionally diploid embryo, and the second sperm unites with 2 haploid nuclei in the female gametophyte, and it creates endosperm. Endosperm, this process of double fertilization is like a major evolutionary step for flowering plants, all flowering plants, dioecious, monoecious. And so this endosperm that develops feeds the growing embryo. The endosperm grows its triploid, and it cellularizes. And once it cellularizes, it can be used by the growing embryo. The endosperm is very sensitive to the ratio or of contribution of the female and the male parent. And ideally you have what's called a 2-to-1 relationship in the contribution of female to male, which works really well. So when you have a diploid by diploid cross, as is maybe nature intended with cannabis, you get this 2-to-1 female to male contribution. You get great endosperm, you get viable seed. In the tetraploid cross, you have a 2-to-1 again. You get good endosperm, you get good seed. You may get less seed than a diploid because you have twice the DNA content in these cells. The process is slower. That's how I rationalize it. But you get ample seed, ample viable seed. At least we do. And we've made the tetraploid cross a couple of times. When you have a diploid mother and tetraploid pollen, the female to male contribution to the endosperm is 2-to-2, and it fails. It doesn't cellularize and it can't be used for growth. When you have the tetraploid mother and the diploid pollen, you have a 4-to-1 female to male ratio, which, as I'll show you, is good enough endosperm. You get more seed set, viable seed, than you do with the reciprocal. So that's here in blue. So after that failed, which was a disappointment for sure, we tried the reciprocal. We had Kentucky Sunshine Tangerine and Purple Star tetraploid parents. Purple Star is an autoflowering type. It is a small plant in the greenhouse. And we used wife feminized pollen, diploid. We had a couple of plants of each. We had much higher percent viable seed, 76% viable seed, 57, and we had 73% viable seed on the diploid by diploid control. So what happened, unfortunately, in this experiment was our pollen parents got dry one day, and we lost 60% of them. And the 40% that remained were weakened. And quite likely our female plants struggled a little. So overall, you can see here, we had much fewer seed, but of those seed, we had many more brown viable seed. I mean, even on our diploid control, wife by wife, we only had 192 seed on a pretty big plant, but 73% of them were viable. And the germination rates, we germinated a subset, were really high on those brown seed, and they were much lower for the reciprocal cross. So it worked better, but you know, we had some setbacks, but the story gets better. We also did have some mixoploid plants. As you see here, we had some diploids, and you'll notice they were with this tangerine cultivar and this purple star cultivar. We had diploids and triploids and tetraploid. And we think in this case, while it could be pollen contamination from a random male flower on, let's say, the Kentucky Sunshine, because this cultivar does that, which is probably why we had tetraploids. We know that autoflowers, although not tested, this is just an observation, we know that autoflowers don't convert to tetraploid as reliably as photoperiod, and we've likely had mixoploid parents here. And that's why we got some diploids. And the same thing with tangerine. We had started out with, I think, 7 different tetraploid tangerines and kept testing and testing. And, you know, we had mixoploids. So that's really something to be watchful for if you're doing this kind of work, which I will get to in a moment, but I know my time's getting close. All right, so those 4N mothers that were mixoploid, maybe early on when we tested them, again, let's imagine all of the white cells in the shoot apical meristem were tetraploid, and maybe there was a cell that was diploid that we didn't catch in an early test, but that cell in this, what would be called a sectorial chimera, grows. If it's diploid, it probably grows a lot faster than the tetraploids because it's got half the DNA it has to copy for each cell division event. And eventually, you know, it occupies a good part of the apical meristem or apical meristems, sections of the plant. And then, you know, if this resulted in a female flower and it gets diploid pollen, you have a diploid seed. All right, so then we're like, okay, this greenhouse is variable in its environmental conditions, especially in March and April, things get really hot. You have an overworked greenhouse crew. Plants maybe don't get watered when they need to get watered. So we tried the cross in a growth chamber and we had really good success. We took this wife tetraploid plant. We used purple star feminized pollen on a pretty small plant. I'll show you a picture in a minute. We had over 1,500 seeds and 65% of those seeds were brown and viable. A very large percentage of those were triploid. And that could be on a saleable scale, perhaps, that many seed per plant. We also used a tetraploid autoflower called Tsunami, which again is going to be an overall smaller plant, which is going to yield fewer seeds just by its size. But we had 500 seeds, 54% were viable, and all were triploid. And all germinated really well. These were the plants in the first growth chamber experiment. These are tetraploid wife plants. So not super big. This is a #2 trade nursery container or a 2-gallon. And they produced a lot of seed. So based on all of this crosses that we've done, we feel pretty confident recommending to go with the tetraploid by diploid cross with as controlled an environment as possible. Also, source that tetraploid parent from seed from a tetraploid by tetraploid cross. So use your colchicine or your rhizoline, make your tetraploids, test it to make sure it's tetraploid, clone it, self it. The resulting seed should be tetraploid, and you only have to test it once. It's not going to be mixaploid from a seed. If it's tetraploid, it's tetraploid. And then use those seed-produced tetraploids as your mother. And this concept of finding your tetraploid and selfing it works well into this strategy that hopefully a lot of growers are realizing, that you need to develop inbred parental lines to produce hybrid F1 seed that's uniform and vigorous for going to the field. I have no doubt that a lot of the problems that growers are seeing with their seed is from it being inbred, from disease to non-uniformity and other stressors. So the next steps have been to test the sterility and performance of the tetraploids. I think I have 5 minutes. And this is what Lauren has been doing as part of her doctoral work. We're testing the sterility using male pollen from male plants because it is higher, it has higher viability, which we've shown and others have shown in the scientific literature. Male pollen tends to be more viable than most fem pollen. This is a video. Oh, look, it's working. So here Lauren is dousing this female triploid with pollen from a male plant in a greenhouse situation. This is the developing seed on the diploid control versus no or very little seed development on the triploids. This is seed collected from one diploid plant from that study. There were over 3,000 seed, many of which was viable. This is the seed collected from a single triploid of similar size. These are viable, these are hollow, these are irregular, malformed. These were cut open to prove they were hollow. Here's some of her early data on this, showing the percent brown and irregular and white. And then this was a separate study to look at performance. We had 4 different diploids. We had 3 triploids. We see some changes that may be significant, like the width of the colas. I do not think this CBD will be significant, but maybe. We intend to do more greenhouse and this summer, hopefully weather permitting, field tests for sterility and greenhouse tests for performance. We have developed crosses that have a diploid cross, the same as a tetraploid cross. Now that my licensing state licensing allows me to make crosses. And this is my last slide. So perhaps slightly enhanced CBD. Our tetraploids have been bigger, chunkier looking plants. And I think some of those traits will be significant. But we are still doing, this is work will be based on the culmination of several greenhouse experiments and field work too. All right, so that's what I have to say, and I'm happy to answer any questions, time permitting.
Fabulous, Jess, thanks so much. There is a couple minutes left, so you get the gold medal for staying on track. If there are questions, go ahead and put them in the chat. Again, we've got a couple minutes before our next speaker. As folks are typing those, I guess I'll ask my question. You mentioned it very briefly. So the phenotypic variation that we'll see in the field, is it, in your opinion, for if it's for triploids, is it more likely unstable genetics or like I think you said breakdown because of inbreeding?
So there are very few companies that are providing triploid seed, and if they have a solid tetraploid parent and diploid parent and they're making their crosses very controlled, they're probably getting a vast majority of triploid seed. It's possible there could be a tetraploid in there, and that one might set seed. If you see a plant among, your triploid lot that's got a boatload of seed, it's probably not a triploid. And that can happen. And hopefully that's not a major detriment. It's not like it's a pollen source. It's just one plant that had seed. But I think the process, if it's done in a very controlled way, can reliably produce triploid seed for market. And the variation again is coming from that this is seed. It's from a sexual source. So there is inherent, variation. We know from the history of agriculture that we want to sell F1 seed to have the greatest uniformity and also take advantage of hybrid vigor. And F1 seed is produced from 2 inbred parents, not just 2 different parents, but 2 inbred parents that were inbred for their traits of interest. So starting with different parents is good, but better to start with parents that have been selfed to the point of at least not losing their fertility. Awesome. Which is where I think, some of the more progressive companies are moving, which is great.
Awesome. Well, I think there are no questions popping up in the chat, and actually we do have a representative from Progressive Company. So we're going to have our next speaker start here in a moment. So thank you so much, Dr. Jessica Lubell-Brand. If you want to go ahead. Okay, perfect. Andrea Garfinkel is the Director of Research and Development at Oregon CBD, industrial hemp seed research and breeding company located out in the Willamette Valley of Oregon. Her work at Oregon CBD involves the discovery of genetic markers for use in breeding new cannabinoids and novel traits. Andrea has several ongoing research collaborations with public universities and government institutions and works to support hemp growers throughout the United States. Andrea holds a master's in agronomy from the University of Wyoming and a PhD in plant pathology from Washington State University, where she specialized in fungal diseases of specialty crops. Andrea is going to talk about understanding and breeding for powdery mildew resistance in cannabis. And I have to admit, this is the most anticipated presentation, at least from my perspective. So I'm going to sit back and get my bucket of popcorn out and listen to what you have to say.
Well, thank you so much for the really kind introduction. That's some big anticipations to fill. Hopefully I can execute. Anyway, thank you again for having me here today. I appreciate you guys inviting me. As Scott said, I will be talking about understanding and breeding for powdery mildew resistance in cannabis. Since Scott already gave me a really nice introduction, I didn't know whether or not I would be on screen. So here I am, and here's our facilities. I work for Oregon CBD. As Scott said, we are located in the Willamette Valley region of Oregon, which is west of the Cascades and east of the coastal region. We're about an hour and a half south of Portland. And just here's an aerial view of our facilities. We are breeders who make seeds. So today I'm going to talk to you about a couple of projects that I've been working on. First, I'm going to talk about pathogen biology and epidemiology of powdery mildew species on cannabis, just to give you a background. I'm also going to talk about some work that I've done on powdery mildew surveys here in the Pacific Northwest. And then we'll go in and talk about resistance genes, and what we're seeing for resistance in cannabis. So with that, just to introduce powdery mildew, it's always so hard giving these presentations because I can't see the audience and I don't know whether or not the audience is familiar with these particular pathogens. But if you are, you know that powdery mildew is potentially, it is the common name for diseases caused by groups of fungi in the order Erysiphales. So there are lots of different species, lots of different genera, but what groups them is several things. First of all, they're what are called obligate biotrophs, which means that they require a living host to infect the tissue. So in contrast to some necrosis inducing fungi that is essentially like kill the host tissue and then eat decaying or dead host tissue, these rely on having live cells in order to survive. So they're not typically killing their host, but they will live on the surface of the plant and have specialized structures that enter the cells and essentially eat and live off of the cellular content. So what you'll see is the signs of the fungus. So the actual fungus itself grows on the surface of the plant, and you'll see this powdery white growth, typically on the upper surface of the leaf in cannabis. You'll also see it along the stems and the petioles, and it can also infect flower tissues as well. So here are just several different images of signs of powdery mildew. And if you zoom in on this just a little bit, what you'll see, I don't know if you can see my cursor or not, but hopefully you can. You'll see this is just zoomed in a little bit. There's this kind of crystalline powdery growth here on the surface.
We see
of the
them.
plant.
Yep.
Oh, okay, perfect. Thank you. So here we're seeing this kind of filamentous type growth. On this image here, we're looking at it kind of from the side, what these little hair-like structures that are coming up off the surface of the plant is the powdery mildew fungus. And if you look even a little bit closer, zoom in a little bit further, what we're looking at is these long strands. These are called conidiophores, and on top of them are conidia, which are the spores of the fungus, growing up off of the surface of the plant. So you can see a couple here as well. They're clear hyaline structures. And again, zooming in a little bit further, here you see a conidiophore with the spores on top. And if you look at the far right here, my image is in there. If you look at the far right here, what you'll see is a spore that's germinated to create what's called germ tube, and eventually it will grow and create mycelium, which is the filamentous sort of fungal body of the fungal body of the organism. And then what will happen is this will continue to grow some hyphae, and then eventually it will create what's called an aprosorium. This is a specialized structure that is under an extreme amount of pressure, so much so that it's able to puncture the cellular surface and enter the cell and create these specialized structures that essentially suck out the cellular components to create the association, the pathogenic association with the plant. So this is just a typical disease structure, disease cycle for powdery mildew on cannabis. We're going to talk about this a little bit, but there are several different species of powdery mildew that infect cannabis. This is what I guess we'll call the hemp type powdery mildew, Golivinomyces ambrosiae. I don't expect you to remember any of these names, but we start over here. What happens is these spores that are created on the top of the surface, or they come in from an external location, these spores, also known as conidia, this is important because fungi often produce lots of different types of spore structures or spore states. We'll see this in a minute, but these are sort of the main driver of epidemics. These conidia will come into the system and land on healthy foliage. They will then germinate to make that germ tube that we saw on the last slide again. These germinate under conditions of high relative humidity, usually around 50 to 70%, and in moderate temperatures at about 70 degrees. These are sort of the ideal conditions. There's a wider range of conditions in which these conidia can germinate. However, it is important to recognize that powdery mildew is different than a lot of different other fungal pathogens. Other fungal pathogens will require what's called free moisture on the leaves, so either a dewdrop or a droplet of water from irrigation. They require sitting in that water in order to germinate. Powdery mildew is different. It doesn't require any free moisture on the foliage surface in order to germinate. So they can germinate on a completely dry leaf under high conditions of high relative humidity in the atmosphere. So these germinate, they grow that mycelium or thread-like structures that we see then colonizing the surface of the plant. You can see here patches of white mycelium, that is the fungal body. They continue in some cases to grow and they can cover the entire leaf surface. And again, what's happening on this leaf surface is, if we zoom in a little bit, just millions and millions and millions of these spores are being produced on the outside of the leaf, and then they're moved throughout the air. They get bumped or just in wind gusts and start the cycle over and over again. So this cycle just continues to reinfect plants in the system. So we're looking at the same disease cycle up here in the right. It's important also to recognize, as I mentioned, there are different spore states of these fungi, and powdery mildew of hemp is one of those that is able to produce a different spore type. So this is what's called the asexual cycle. All of these conidia that are formed are asexual propagules. However, there is what's called the sexual cycle, or sometimes referred to as the overwintering cycle of this fungus, in which case this is happening throughout the growing season. And then when conditions become unfavorable for the fungus, maybe it gets a little colder out, it produces these specialized structure known as chasmathecia. You can see them in the slide down below. These sort of like orange, reddish, brownish speckles on the leaf surface. These are essentially hardened, melanized, meaning pigmented structures that contain ascospores. So this is another type of spore state of the fungus, and they're contained within asci. You can see it here. So what you're looking at here is chasmathecium that's been broken up, that's been smashed to release the asci and ascospores within them. And so when conditions become favorable again, like in the springtime, these chasmathecia will open up, they'll germinate, they'll release the ascospores from the asci, and then here start the cycle, land on infected foliage. they'll germinate. I don't have a picture of a germinating ascospore, but again, just like the canidia will germinate, these ascospores will germinate and start the cycle all over again. It's important, I guess, to note that the sexual cycle hasn't been observed in most of the locations where hemp powdery mildew has been, where it's been reported. The only place that the sexual cycle has been observed on hemp is in Kentucky. The sexual cycle is important because this is a persistent living structure that can survive over the course of the winter time. So if you have foliage, infected foliage that falls in the field over the winter that has these chasmathesium, they'll start the cycle of the disease cycle early on again in the spring. Whereas if you don't have the formation of these structures, you're waiting essentially for these spores, these canidia, to come in from an external site in order to initiate the disease cycle. These canidia are very short-lived, maybe 24 hours. So the sexual cycle is important epidemiologically speaking. So I was just showing you sort of a general disease cycle of what we're calling the hemp powdery mildew, Gallivanomyces ambrosiae. In the literature, you also see it referred to as Gallivanomyces spadius or Chicharium. Fungal taxonomy is totally crazy and changing all the time, but this is the most recent name of this species. It's also, it's not very host specific. A lot of powdery mildew species only infect one host species. Gallivanomyces ambrosiae is mostly found on plants in the family Asteraceae. So this is your daisy family, but also able to infect hemp as well. And then there's a second species of powdery mildew on hemp called Podosphora macularis, which is more host specific. It's known as the hop powdery mildew. As some of you may know, and I think that Jess alluded to in her previous presentation, hops and hemp are really closely related to each other, or at least as closely related to each other in terms of they're both in the same plant family, Cannabaceae. So hop and cannabis are the most closely related to each other in terms of economic crops that we know of, or commercial crops. So it is interesting that this Podosphora macularis appears to be showing up on hemp too. Something important about these two species, you can't really distinguish them from a macro scale. So you can see here on the left, Gallivanomyces ambrosiae, the white powdery growth on the surface. On the right-hand side here, Podosphora macularis. Maybe this particular sample is a little fluffier. It could be the conditions in which it was grown, but typically it's hard to determine from just looking at the plant from this scale which species you're working with. You have to look kind of at the spores or maybe at the DNA. You can see at least morphologically, the fungus Gallivanomyces has these sort of barrel-shaped spores, whereas Podosphora macularis kind of these rounder, more oval-shaped spores. So both of these, as I said, both of these two groups of fungi have been, or both of these species of fungi have been reported on cannabis. Most of the reports are of Gallivanomyces. You can see here just a couple of publications citing the finding of this fungus. So up in the upper left-hand here side, 2018, it was found in Canada, then in Kentucky in 2019, Ohio 2020, Oregon, in Israel, in New Jersey. This is such a cosmopolitan fungus. It's likely to be in all of the places where hemp is being grown. And something interesting about the Podosphora macularis, and we'll talk a little bit about why we're interested in these two species in just a second, but because there was the previous knowledge that hops is the most closely related economic plant of interest to hops, there was some interest in understanding whether or not the hop powdery mildew could infect hemp because of the increase in hemp acreage, especially around hops production. So there was a study done in New York in 2020 that asked this question: can the hop powdery mildew infect hemp? At this point in time, they hadn't found it naturally occurring in the field, but they took this fungus and intentionally tried to inoculate or get hemp sick with the fungus from the hops, and were successful in doing so. So this was sort of the first initial clue, I suppose, in figuring out whether or not this organism could infect hemp. And then subsequently, last year, a team of us working here in Oregon were able to find this Podosfera macularis, the hop powdery mildew, naturally occurring here in Oregon. And then also Zamir Punja, who's done just a tremendous amount of work on pathogens of cannabis in BC, British Columbia, Canada, also found it naturally occurring in the field in Canada. So we wanted to know what the frequency was in the Pacific Northwest of these two pathogens in hemp production in the Pacific Northwest. And again, I was kind of alluding to this fact that, it's potentially important because they're closely related to hops. But maybe what you don't know, if you're not from the Pacific Northwest, is that the Pacific Northwest is a huge producer of hops. And so is New York, which is why they were interested in the first place. And in a lot of cases here in the Pacific Northwest, what we see is that hemp is being grown either in the near vicinity or even immediate vicinity of hop yards. And you can see here, I wish I had a better picture of this, but I don't know if you can see at home these trellises along the sides of this hemp field here in the back. These are hop trellises. So this is a hop yard directly, there's a hemp field. It's functionally right in the middle of two hops yards. So certainly if there is any cross-infectivity of these pathogens, we would expect to see it in areas like this. And just to sort of hit home the point, like, why do we even care about this particular pathogen in the Pacific Northwest? And part of it is because Podosfera macularis, this hop powdery mildew, is an actual quarantine pest here in the Pacific Northwest. This is because it's highly destructive of of hops and of huge economic importance. And at this point in time, the movement of green hops material in and out of the tri-state area, which includes Oregon, Washington, and Idaho, is restricted. So you have to make sure that any movement of hops in and out, or especially into this tri-state region, is free of this pathogen. So the question is, if we have the hop powdery mildew showing up on a whole new host, it's not just hops anymore. What does this mean for quarantine policies here in the Pacific Northwest? Is this a threat to both industries? We needed to have a better understanding than just looking once and finding powdery mildew on, just an off chance sighting of this particular pathogen. So what we did, quickly before I go there, we have here an image of Washington and Oregon. This is just sort of showing what we knew prior to any of the surveys that were being conducted. So these are county maps of Washington and Oregon, and in the colored counties, we have different species of powdery mildews that had been reported previously. So we know in green, Podosfera macularis, the hop powdery mildew has been had previously been reported. In white, Galivanomyces ambrosiae, this is the quote unquote hemp powdery mildew. And then in the counties where you have this dark color, both mildews had been previously reported. But of course, this isn't representative of all of the growing regions. And a lot of these data were taken prior to the explosion of hemp acreage here in the Pacific Northwest. So you can see these cones are representing where there's major hops production, and the little cannabis leaves are where there's, hops production. I don't know if I misspoke. Hops production of these little cones, cannabis leaves is hemp production in each one of these states. So clearly there's a lot of the state that's not being represented. And also these are sort of older data. So before I go on a little bit further, I just want to mention that this work was done in collaboration with the USDA ARS and Oregon State University. The PIs were Dave Gent and Cindy Oakham. We got a grant through the Western IPM Center to conduct this research. Hannah Rivedal of USDA ARS was also really highly involved in this project. And I just, as well as these people, and I just want to give a quick shout out to Michelle Wiseman. She produced a lot of the figures that I'm using in this presentation today, and I want to thank her tremendously for helping out with that. So we had three major questions in this research. First of all, where is the powdery mildew present? When is it present? So we looked at three different time courses throughout the growing season, early, mid, and late season. And of course, we wanted to know which species are showing up in which state at what time. So what we did is we walked 2 transects in each field. It's kind of a fancy word for row. And we looked at 100 plants per transect, and we collected any foliage that had signs of powdery mildew on them. We took them back to the lab and we extracted DNA and did DNA sequencing to identify the pathogen. And then we did some further characterization using a process called qPCR in order to determine some additional questions about the specificity, especially like mating type and virulence type of the especially Podospora macularis samples. So here's just a quick image of the sampling sites in each one of the locations. We had 12 sites in Washington and 21 sites in Oregon. I guess I should also mention that this Western IPM grant that we received, it was geared a lot towards understanding what the risk is for the hops industry. It's a lot of how we justified getting these fundings. Dave Gent actually on the grant is a hops guy. So we focused a lot on hops regions because we also figured these were the highest likelihood of finding the hops powdery mildew. So you'll see, even though we had that huge wide range of sites where hemp is grown in each one of the states, the Washington sites were especially focused because these were focused in what's known as the Yakima Valley, which is the major hops growing region of Washington. And then in Oregon, we have a little bit of a wider range. We focused a lot of the samples here where I'm located in the Willamette Valley, because that's where most of the hops production is here in the state. And then also we hit a couple sites further south as these are major hotspots for hemp production in the state. And we also, thanks to a collaborator of ours, received one sample from BC as well. So sorry, I know I'm cruising super quick through all of these, and I'm gonna go through our data really quickly because I bit off more than I can chew. I wanna tell you guys about all the things that we're doing, but I just wanna, these next couple slides, what you're seeing here is these little bubbles are representing how many samples that we got from each place, from each sample site. So the bigger the bubble, the more samples that we've had. In this particular instance, the color of the bubble is indicating at what sampling point we found these species. So as I said, we had one sample from BC from the late season. And in Oregon and Washington, something that was interesting and kind of expected to us is that while we were sampling early on in the season and in the middle of the season, we were only getting like 10, 20 samples in Oregon from each of those 2 sites, none in the earlier mid sampling time points in Washington. And most of our samples were coming in the late season. So when I say late season, I mean like September, October. In Oregon, we were collecting 107 samples in those late seasons. In Washington, just 9. So it wasn't that many this year, but we were seeing these powdery mildew infections occurring later in the season. And I'm just going to show you quickly, this is temporally and spatially what these samplings looked like. So this was early June and July. As I said, we're only finding a couple samples here in Oregon, in the Willamette Valley. These were all the hemp powdery mildew, Galliminomyces ambrosiae. If we look at August, still the only places that we're finding powdery mildew is here in the Willamette Valley, same species, the hemp powdery mildew. But again, we're looking at very few samples. And then we start to come to September, and all of a sudden powdery mildew starts showing up a little bit more. So this is considered our late season. We're seeing a lot of the hemp powdery mildew show up in the Willamette Valley, as well as further south in like the Rogue Valley area of Oregon. And then our one sample shows up in Vancouver as well in this late season. And in October, this September-October distinction is a little bit misleading because we didn't look at the same locations in both September and October. They can kind of be considered late, kind of collectively. But it was in these October sampling points that we started to see even more powdery mildew in not only in the south, the Willamette Valley, and also here in the Yakima Valley region. And lo and behold, Podosfera macularis in these, the hop powdery mildew in these hop growing regions did start to pick up quite a bit. So just kind of a takeaway, you'll see ultimately, you know, for as much sampling as we did, most of this, the powdery mildew species that we found were the hemp powdery mildew, almost 92% of the total samples. Just a small fraction of the total samples that we found were the hop powdery mildew, so just 8.6% of the total. And again, we're just seeing late season being the most important, at least in 2021 for powdery mildew. And I think that, you know, if we look at all of this collectively as just looking at these takeaways here, it's just important to recognize that we have 1 year of data. These surveys will continue. And this year was especially hot and especially dry, which means a low pressure year for disease for powdery mildew. And I think that we have a lot of ongoing questions about what will happen as hemp acreage decreases in the state. So we had, and I think that this is nationwide, we had a huge explosion of hemp acreage and now a huge contraction, you know, where these fields move, the sizes of these fields will all have an impact on how the disease develops, and which species, you know, we'll continue to see. I think still it's just unclear to us really what concern powdery mildew is to the hemp industry in the Pacific Northwest. But I know this is a lot of data to just say we don't know what the impact is, but I think understanding sort of the early onset of any epidemics is going to be critical in tracking the progress of these diseases in these newly emerging industries. And of course, you know, I've presented these two species, and I think probably growers are like, why should I care? You know, why is this important that there are two different species? Not only could these be managed differently, the epidemiology of them could be different, but also I think we have some ongoing questions about whether or not quarantines are going to be important here in the Pacific Northwest if we continue to see the hop powdery mildew occurring on hemp. That could really restrict movement of green plant material of hemp in and out of the states, which will be really important for hemp growers moving forward. And you know, as I said, ultimately, what will the challenges be of managing these pathogens into the future? I think just continuing with these surveys and understanding what we're up against is going to be able to help us better answer these questions for growers. But with that being said, I want to move on to the final. Yeah, please, Scott.
You're good. I was just going to let you know you had about 10 minutes. So
Thank
perfect
you.
timing.
Thank you. Oh my gosh, I really hope so. I'm cruising, I know. So I'll briefly talk to you about resistance genes and breeding for resistance. So we're talking about managing powdery mildew. And when we're talking about management of diseases, one of the most effective, or one of the tools in your tool belt for managing diseases is breeding for resistance and having resistant plants. So I want to introduce this concept of resistance, what resistance means. These definitions are from the American Phytopathological Society, and being resistant is having or possessing properties that prevent or impede disease development. And this contracts with susceptible, which means that a plant is able to get the disease. And I just want to distinguish this from tolerance, which is the ability of a plant to endure an infectious, or in this case, infectious diseases that we're talking about, without critical yield loss. So resistant means that the plant isn't getting sick, or it's only getting sick a little bit. Tolerance means the plant is getting sick, but it's not having significant yield impacts. There are several different types of resistance, and not all of them are complete. So not all of them are yes, no, yes, it's getting disease, or no, it's not getting disease. Plants can also have what's called partial or quantitative resistance, where you have varying levels of disease. So a plant may just get a little bit of disease, so it's a little bit resistant, all the way to, so it's more resistant or less resistant. Resistance in general is governed by genes or a set of genes, or can also be determined by other aspects of the plant characteristics. But today what we're going to be talking about is resistance governed by genes, especially what are called resistance genes, also known as R genes. So R genes are genes in the plant that encode proteins that detect pathogens and signal a counterattack. And there are lots of different classes of these R genes. It's not important except to know that there are lots of ways to make resistance. But what's critical about these resistance genes is that they're very specific, essentially gene for gene interactions where you have virulence or the ability of a pathogen to cause disease, genes that regulate a pathogen's ability to cause disease, and genes that cause a plant to be resistant, that interact with each other on this very specific molecular level in order to create a resistant plant. And typically, because this interaction is so specific, this gene for gene reaction, oftentimes these genes are very specific to a pathogen. So if we see a plant that has a resistance gene for powdery mildew, even if we see that it has a resistance gene for the hemp powdery mildew, it may or may not work against the hop powdery mildew, for example. So very specific interactions. And when we're talking about our genes specifically, usually we're talking about genes that confer this vertical, complete, or qualitative yes/no type resistance. Does it get sick or does it not get sick? It's not this sort of gradation of sick. These are usually heritable, these genes, and they're usually dominant. So plants only need one copy of them in order to be resistant, and which means that they're easy to integrate. So they're easy to introduce and bring into a breeding program and bring into new plants, which means that these are the types of resistance genes used most commonly in major cropping systems as a pest management strategy, due to the ease of which they can, the effectiveness of them and the ease of which they can be introduced into breeding programs. So what does an R gene look like in cannabis? Here's an image here. On the left-hand side, you see a susceptible phenotype. This plant has been grown in high humidity conditions with a lot of fungal spores of the powdery mildew pathogen. And you can see its leaves are just completely covered in powdery mildew. On the right-hand side, you see an example of a resistant phenotype that has no mycelial growth on the surface of the leaf. These plants were grown side by side in the same conditions as each other. It's not that they've been excluded. They look separate because they're two different panels, but these plants grew right next to each other in the same environmental conditions. One got sick and one did not. Clearly, this is a very strong R gene that is able to detect and fend off attack by the hemp powdery mildew, Galliminomyces ambrosiae. So this image is from a paper, and this paper was a result of a collaboration between me and another scientist at a cannabis company located in Pullman, Washington, Dewey Scientific. We were able to essentially discover the first R gene in cannabis, which was really exciting for us to do. No R genes have been described since. So we're really looking forward. This is a just newly emerging field of science in cannabis, and a really exciting step for the industry, because this means upon discovery of these particular R genes, we can then breed for resistant plants and bring that to the market for growers, so they don't have to worry about managing these pathogens. And just quickly, I think I have 5 minutes. So I just want to quickly describe this process of determining how we, discussing how we determine resistance and how we essentially use resistance in the breeding process. We have a susceptible parent, we have a resistant parent, we cross them. All of the progeny will be either susceptible or resistant. We use molecular tools in order to sequence the DNA from all of these different progeny. And we essentially look at all of the DNA and say, where are the differences in all of these locations in the DNA? And which are the differences in the DNA that describe this difference in phenotype, this resistance or susceptible phenotype? And through some quantitative analyses, what we're able to do then is actually physically locate the gene in the genome. In this case, we found a gene on the second chromosome of cannabis way here at the end. And physically finding the gene, you may say, who cares, so what? Well, once we physically found the gene, we can then produce a set of molecular markers that allows us to quickly screen at the very seedling stage without ever exposing any of these plants to powdery mildew. We have the ability to detect which plants have the gene for resistance and which ones don't, and move those quickly through the selection process, and therefore quickly breed for powdery mildew resistant plants. I just want to mention one other paper that's talking about resistance, powdery mildew resistance in cannabis. This is a paper that came out in September of last year talking about what are called MLO genes, or mildew locus O genes. And instead of being resistance genes, these are actually susceptibility genes. So if you have a fully functional MLO gene, this means that you have a plant that's susceptible to powdery mildew. Having non-susceptibility genes, or the absence of these susceptibility genes, can also help breeding for powdery mildew resistance. So in the end, there's just a lot of exciting research. I know that that was a lot in a short amount of time and a lot of preliminary stuff, but ultimately we're making headway in figuring out how to bring bigger and better plants to the industry. And that's it.
Awesome. Thank you so much, Andrea. Actually, for those of you who weren't around this summer, we had folks from Oregon CBD out, and Andrea dropped a little teaser about this research. And so that's why I was so excited to hear this. If you have questions, John Jemison has posted twice how incredible, first the pathogen and now your presentation. So thank you, John, for the accolades. Again, we've got a couple of minutes for questions. I've got a whole list of them here I can ask, but I can always email Andrea separate. So if there are questions, go ahead and put them in the chat. Otherwise, I'm just going to ask all of mine in the next couple of minutes.
Go for it. And I can also put my email in the chat in case anybody wants to follow up as well.
That's perfect. And actually, if questions come in the chat during the next session, you could also answer them in the chat. Either way will work. So yeah, like I said, I have a whole list of them. I'm not going to barrage you with them. So I think I remember last year at our hemp conference, the keynote speaker talking about resistance to powdery mildew, kind of being restricted to just varieties that were a high THC cannabis. Is that, do you know if there's the same trait in what we call industrial hemp, the low THC varieties as well?
That's a great question. I don't know that at this point in time. So this research, well, maybe I should hesitate to say it on a recorded presentation, but this particular research was done at Dewey Science. Like some of these analyses, like a lot of what I did was support from afar. These were high THC type cannabis. However, the nice thing about these particular genes is because they're single genes, they're really easy to just move over just in a single cross over to a hemp background. That's not going to be, well, I guess it would take two to get it high CBD, but I don't know whether or not this exists in CBD populations. I do know that there's some work out of Cornell being done in high CBD cultivars that have indicated that there is some amount of resistance there, probably qualitative and not our gene-based resistance. But I do think that there's the potential that it's out there and we just haven't seen it yet. So growers, if you're out there and you see a whole field full of powdery mildew, but you have a couple of plants that don't have it, you may have the next R gene candidate.
All right. Well, then email me and then we'll start our, no, I'm just kidding. Thank you so much, Andrea. That was awesome. And we have one more stellar presentation coming up in this session before we break for the day. So I believe John Bruce is hanging out there in the background. So John, can you share your screen? So if you don't know, John is a friend of mine and a colleague on the Northwest Crops and Soils team. He's an extension research specialist at UVM. He focuses on applied hemp research. and works with hemp growers on a variety of agronomic topics. He also leads up a bunch of trials in a variety of other crops. Without further ado, I'm going to let John talk to you all about our Northwest Crops and Soils Program 2021 variety trial evaluations on triploids. So John, if you want to take it away.
Great. Thanks, Scott. Yeah, so Scott knows how much I love doing presentations too. So this is going to be fun. So I've been working with a team for about 5 years now, maybe a little longer. So I've focused mostly on our hemp research trials. I've also done a little bit of work in hops, small grains, and other crops that we work on up at Borderview Farm. So I think this year is going to be my 4th year working with hemp. So I'm still definitely learning a lot as we go as well. And then I guess kind of keeping with the theme here, I'm just going to talk a little bit about our variety trial and some of the differences we've seen across all these different varieties, including some of those commercially available triploid varieties, and then some of those responses to powdery mildew and other diseases. So we end up looking at a lot of different things for these trials, like growth habit, pests, yields, and chemical compositions as well. So we're really just looking to see how these varieties perform in our climate that are, really bred in different parts of the US. And also, like, if anybody's interested in kind of digging into this stuff a little bit more, we do have all of our research reports posted on our website as well. And again, so for anybody who's not super familiar with our program, we do try to cover a pretty wide area of topics for our hemp trials that are focused on developing those best practices for hemp, just like any other crop. And so these are really broken down by kind of our end product, which includes, all of our different flower trials and grain and fiber trials as well. And, of course, for these purposes, I'm just going to focus on our hemp flower variety trial today. But yeah, we just thought it'd be good to give a little update on everything that we've been doing up here in Alberg. And, of course, every, each type of hemp, depending on the end product, has its own unique challenges. So we're really just trying to cover a lot of ground in as short amount of time as possible. And so since I'm just going to focus on 3 years, basically. But so since 2019, we've had, between 20 and 30 different varieties each year that were entered into our trials from breeders across the US, which is mostly included full term or photoperiod sensitive varieties. In addition to a few autoflower varieties in these most recent 2 years. And those are mostly coming from distributors in Oregon and Colorado, and some from South Carolina and Vermont as well. So we try to gather, varieties from as many different areas as possible with folks that are willing to enter those into the trials. And we evaluate them really just based off of the plant material that were provided. So we end up evaluating those varieties that are either grown directly from seed or rooted or unrooted cuttings. Though in the past couple of years, we've really kind of transitioned to seed because those kind of more directly reflect what a lot of growers are putting out in the field. And so within these trials, so I guess as I go along, I'm just going to touch on like a few of those different metrics that we collect. And then a few, of course, a few of those are going to include, the plant characteristics like height, width, whole plant weight, and then the fractionated components of the plants as well, in addition to our time to maturity. And so for our trials and in our region, this will probably differ a little bit based off of where you're growing. But We try to shoot for planting date that's going to be within that first or second week of June. Sometimes that's been a little challenging with our trials, especially I think it was in 2020 when we had some shipping delays, where some of those ended up getting planted, almost a month later. And then those also kind of end up having some pretty stark differences in yields when compared to those that are able to get in June. But in general, we try to have everything in the grounds in those first 2 weeks, because that delay really shortens the vegetative period and then overall plant growth. So we want to try to get something that's as representative as possible of the potential for those varieties. And then throughout infield growth, we're monitoring a lot of different things, but one of those main ones being flowering week. So we're interested in seeing, when each individual cultivar initiates its flowering sequence, and then if there's any variability within that single variety. And then, as you can see, we also look at, whether or not, based off of, our harvest week, if those varieties are going to mature within our given growing season as well. And so these two pictures here are kind of looking at two different varieties. And so this one on the left here is showing a few that were grown from clonal propagation. And you can see there's a much more uniform growth habit with those compared to this one on the right, which is these two right here next to each other are of the same variety. And there's almost a 3-week difference in flowering dates and maturation. So there's, scrolled a little bit. So there can be a fair amount of seedling variability. So that's something that we're also kind of looking at and monitoring as we're going along. And as I mentioned before, there's we're also growing grain and fiber varieties at the farm. I'm not including any of this or any of that information in this presentation, but there's a pretty it's kind of interesting to see flowering dates between some of the male plants within those grain and fiber trials, and differences in the flowering times within our flower variety trials. And it seems like a lot of the pollen actually doesn't have a chance to fertilize those flowers based off of a time difference in flowering. So that's been kind of interesting to observe as well. And so Scott's going to talk a lot more about insect pests tomorrow, I believe. But I just wanted
as much as I could.
But so I'm just going to touch very briefly on a few of the things that we've seen in our variety trials as well. We've also been doing this. So part of what Scott's going to talk about tomorrow is this Vermont and multi-state scouting or pest survey. But so a few of the things that we've seen at our research farm have been flea beetles, corn borer, earworms, aphids, and leafhoppers. And so this is observed through, part of our weekly scouting for all these different pests. And as a whole, at least from what I've seen within our trials, a lot of these don't seem to have too much of an impact on the, end crop or end product. We see a lot of skeletonization and just general leaf feeding through or as a result of the flea beetles. Definitely seen like a little bit of hopper burn, mostly on some of these earlier maturing autoflower varieties. But as a whole, those two don't really seem to have too much of an impact on our crops. But I guess that's something that might become a little bit more of a problem moving forward, depending on, overall hemp acreage, or, weather conditions, various other factors. And, of course, hemp borers also, they seem to have, a little bit more of the, or more shocking impact on the plants in earlier parts of the season. And in worst cases that I've seen personally, these can kind of take plants down right at the base. But for the most part, it seems like most of the plants are able to really recuperate and kind of grow through this type of damage. And then, so here are just like a few more pictures of hemp borer damage. So a lot of these weakened stems can lead to some lodging, but a number of varieties have also been more or less susceptible to wind damage over the years that we've noticed. And the variety on the right is actually, I think it was originally bred for indoor cultivation. So that's one that, really struggled to support itself in the field, especially once it started to flower. And the majority of the plants that we've had in our trials have had pretty good resistance to lodging. So a lot of those haven't, shown any of, the toppling or the broken branches. And many of them have even withstood some pretty severe windstorms. And up at Border View, it's always windy for anybody who hasn't been up there. So it's definitely a good trialing ground for a lot of these in terms of structural stability. But we've also seen a good amount of differences between those that have been started from seed versus those from cuttings, which it seems to be mostly as a result of, root growth. Some of those seedlings having much more extensive root systems. Let's see if that goes. And again, part of our weekly scouting, so we do often look at powdery mildew, leaf spots, gray mold, and white mold, which have been some of the more common diseases. Oops, going back. And, compared to that first year in 2019, when we really started to evaluate disease pressure, the past 2 years, we've definitely seen a buildup in disease, which is, maybe just a part of increased cultivation in our area and just general disease pressure buildup in our fields. But it seems like, at least up in the islands where we're growing, we do see a lot more leaf spot and powdery mildew and white mold. And then, a little bit later on in the season, especially if we're seeing, some physical damage, increase in gray mold pressure as well. And so then this here, I just thought was kind of interesting. But this is a picture of the same hemp variety side by side. One of which was severely impacted by leaf spots, and another that, really wasn't, it seemed like it was untouched for the most part, maybe a few instances here and there. And that's not something I'm super familiar with, but I thought it was interesting within varieties. And so maybe that's something Andrea or Jess could talk to a little bit more. But this was actually at a farm in South Hero, which was part of our scouting project. But it seems like there's some pretty big differences, in that resistance and expression from those seedlings in a number of different ways, other than just like the flowering times that we've seen in some of our trials at the research farm. And in addition to that, we've also seen a lot of resistance in, I guess, just general disease buildup, especially this last year in some of those CBD cultivars. And in addition to some of those that we received from South Carolina or Colorado, which had a much thinner leaf structure. And then comparing that to some of the CBG varieties, which appeared to be a lot more susceptible to powdery mildew. I think there were 3 different ones that we saw a lot higher pressure in there.
You got about 10 minutes, John.
Thanks, Scott. And then, so this is going back a little bit to our harvest week and maturity as well. But frost damage can also be a pretty major problem for growers in our regions as well. So it's, first off, really important to know your hardiness zone, especially in these northern climates, and to figure out, what that growing window actually looks like, especially for hemp. But in some cases, it might be better to, have a crop that's able to mature within a timely fashion and not get kind of pushed out into, mid to late October for us, especially for some of those folks kind of growing up in those mountainous regions, which I know there are, a fair number of. And so in that case, of course, like the early maturing varieties are, might be a better way to go to avoid any of those problems. And then this is actually from 2020, but any of these ones that were highlighted, I think about half of them were actually from that late planting in July. So that's, again, really important to, make sure that you're getting that crop in a little bit earlier or as early as you can if you're, hoping for it to reach, a full and mature size. And so for these next few slides, I'm just going to look at, or just kind of go over like some of our basic methods. So everything that we do, we kind of do on, or the measurements that we take are on a, wet basis. So we're taking the plants out of the field, cutting them at the base, and then weighing the whole plant up. And so this kind of helps to give us that, those fractionated components of the plant. And so we're breaking them down into individual branches, removing fan leaves, running them through our bucker here, and then running those through our trimmer, which these are just some little quick, I don't know if they're going to play or not, but it's not super important. But yeah, so we're just running those plants or that flower material through our trimmers. And then all of that material gets weighed up afterwards. And so then that gives us this really fun and exciting chart, which I'm just going to gloss over a little bit, but a few of the things, so again, like we're gathering that whole plant weight, then we're taking individual components of that. So we're gathering that stem weight, flower weight, leaf weight, and then the proportions of each. And the whole point really behind gathering that information is to, kind of look at a little bit of, potential labor inputs, differences in, the proportions of flower material versus stem material. And then, eventually we'll probably work on developing, nutrient recommendations and crop removal rates based off of, those different components and what's being taken out of the field. And then, so again, just kind of looking very briefly at some of these yields. Actually, I just want to go back really quickly because this past year we had a few of those different commercially available seedless or triploid varieties. And for the most part, I think visually the triploid plants like appeared to be much larger, much taller in general. And then it shows that, some of those weights, those whole plant weights were a bit higher as well. And then I think, I'm trying to think of in the flower material as well. But for the most part, they were, fairly similar, comparable. The plants visually were a good bit larger, which I just thought was interesting as well. And then kind of, again, going over some of these different yields. So across the 3 years, we've seen, pretty wide range. And these are dry matter flower yields specifically. So we're not looking at biomass, though we could. But so those have kind of ranged from, 500 to, nearly 4,000 pounds per acre. depending on the year and the varieties. And everything in our trials are, they're all planted at 5-foot centers, with the exception of some of the autoflower varieties, which we've kind of adjusted those spacings to 2-foot centers, just to really accommodate for that, those plant growth metrics, or just kind of thinking about the potential for reducing plant spacing to maybe increase yields, and that could also lead into if you have a delayed planting and you're expecting plants to not reach that full size, maybe you could plant those at a little denser plant spacing. But you still also want to keep in mind some of those potential for disease pressure and maintain proper airflow within the field. And
You had a couple minutes left, John.
thanks, Scott. And so I think this is kind of getting into a little more of like the nitty gritty stuff, and potential concerns moving forward as well. And sorry, it's such a busy graph here too, but I just kind of wanted to put everything together from at least these past 3 years, and I think this is looking at all 65 varieties that we've had entered into our trials. Some of these have been trialed for 3 years, some just 1, but I think in general, like our at our research farm, our cannabinoid levels are pretty average when compared to some of these specialty growers who commonly reach upwards of 20% CBD or other cannabinoids for their crops. But we generally saw a number of varieties that were well above that 10% range for CBD or CBG, but they also had THC levels that were above this 0.3 limit that's in place. So of course regulations are definitely going to be challenging moving forward for a number of folks, but we can kind of pick out, I think it's a little less than half of these varieties that if they're grown to full maturity, they're still kind of maintaining something just below 10%, but also below that 0.3 limit. In addition to some other potential options for harvesting at a little bit earlier date or testing at a little bit earlier date. But I figured it would be good just to kind of bring all of this together, just to show what we have found. And then for anybody, I think Stephanie Smith is gonna give a great presentation tomorrow, just for like a little bit more of an update on some of those regulations, which will hopefully provide a little bit more clarity in some of those regards too. And with that, I think I'm wrapping up. So if anybody has any questions, give a shout.
Awesome. Thanks so much, John.
All right, one from John.
Yeah.
So he said, did you find it harder to get the triploid to maturity compared to the diploids? So I think honestly too, I think that we might have harvested those a little bit earlier than we should have. I did personally notice, I think it was maybe like a 1 to 2 week difference in flowering times between the similar varieties that were either diploid or triploid. I'd be curious to hear a little bit from Andrea on that too, if she has any input, or if that's something that they've noticed as well. But it did seem like they were a little bit further behind. So I think our harvest could have been pushed out on those a little bit further, if we had, been anticipating that a little bit more.
And then there was also a question about seed. Did you guys look at seed?
Yeah.
In the diploid versus triploid?
So we actually planted those triploids directly in our grain trials too. which is obviously not something that you would normally want to do. I don't think we haven't finished cleaning all of those flowers, but we definitely saw a seed set. I think it was similar to Jessica's findings in some ways, that those seeds didn't fully mature, and were actually hollow. But we haven't done any germination tests on those, or as thorough of a breakdown.
All right, well, we will wait to see those results.